Welcome to the video on the ribosome function and ribosome physiology. So probably by now what you know is that ribosomes are protein synthesis factories, okay? And this video is going to be dedicated to how the ribosome works. Now, you're looking at this in the context of anatomy and physiology. Perhaps you're even looking at it in the context of biochemistry, molecular biology, whatever. And what you might notice, if you're really observant, is that this is 50S and this is 30S. Now, that is bacterial. Okay, that's bacterial. Okay, so this is a bacterial ribosome. This is not a human ribosome. Okay, now you ask, why do I show you a bacterial ribosome? Well, this is going to be the mechanics of the bacterial ribosome, but what you would find is that eukaryotic ribosomes work very similarly. Okay, the proteins are going to have different names, but in general, the mechanics are going to be pretty much identical. Okay, so what we're assuming in this video is that we're starting off with several things. Okay, let me orient you with this. Okay, number one, this is the mRNA. This is the mRNA that we've talked about in great detail that we made during transcription and mRNA processing. And we have several things associated with the ribosome. Number one, I have something referred to as the A site. A stands for aminoacyl. Okay, we'll talk about what that means in a minute. I also have this P site right here. P stands for peptidyl. And then the ribosome also has an E site, which stands for exit site. Now, there is one thing I want to be absolutely specific about. Okay, every cycle of the ribosome begins with either one amino acid or a growing peptide chain attached to a tRNA in the P site. Okay, so when you start translation of the mRNA, okay, notice I have a codon right here which is AUG. Okay, essentially AUG codes for methionine, and that's usually the way proteins start. And of course you have the anticodon of the tRNA, so I'm circling the tRNA in red. So there's the tRNA, and then I'll do this methionine in green. Okay, there's your methionine. So basically, I'm starting with an amino acid. I'm starting the cycle of the ribosome with an amino acid or a growing chain attached to a tRNA in the P site. That's important to realize. That's the start of each cycle. Okay? At the start of each cycle, when I have that condition satisfied, any other aminoacyl tRNA has to enter to the A site. Okay? So assuming I start off with this condition, so this is the start of the cycle of the ribosome, like this in both eukaryotes and prokaryotes, okay? The incoming aminoacyl tRNA always has to enter into the A site. Thus, it's the aminoacyl site. So here's my incoming aminoacyl tRNA, okay? But see, it's not as simple as you think. In general, the incoming aminoacyl tRNA needs a little bit of help. And it's going to get that help through a G protein called 2. So literally what 2 does is it facilitates the entry of the aminoacyl tRNA into the A site. That's literally what it does. And so what you end up getting is, again, you have your initial condition where, I'll do this in red like I did before, you have your initial tRNA. You know, there's your initial tRNA with, initially this was methionine, or it could be the growing polypeptide chain. And now you have a tRNA, which I'll do in blue. Actually, let me do it in black because I did that before. So you have this new tRNA in black, and then this new amino acid, I'll do this in blue. Here's your new amino acid. And now that is in the A site. Okay? Now here's the important thing to realize. So let me specify this is the blue amino acid. And then, of course, this one is the green amino acid. So here's what's going to happen. Okay? The amino acid, or any growing chain of amino acids on the P site, are going to be transferred onto the amino acid in the A site. So literally, this amino acid is going to attack this amino acid or peptide, cleaving this bond right there. Okay? So notice what happened. Okay? Here is the blue amino acid, okay, that was originally on the incoming aminoacyl tRNA, and the green amino acid, okay, essentially is going to get transferred onto that amino acid, okay? If, for example, let's say, for example, there was a black amino acid here, let's say there was a red one there, maybe there's another blue one here. Okay, if that were the case, then you would see the same thing right here. Here would be a black amino acid, here would be the red amino acid, and here would be another blue one. Okay, so basically what I'm saying is that, whatever is on the tRNA and the P site, the either one amino acid or growing peptide chain, all that business gets transferred onto the amino acid in the A site. And hopefully you see that here. Now, this particular reaction is catalyzed by an enzyme, which we'll see on the next page. This enzyme is called peptidyl transferase. Okay? And this is an unusual enzyme because it's not a protein. Okay? This particular enzyme is made of ribosomal RNA, or rRNA. Now, we haven't really talked much about rRNA, and you usually don't, but I'll mention it here. rRNA is synthesized through transcription, in basically the same way that mRNA or tRNA are made. Okay? But this particular type of transcription is basically done by an RNA polymerase in a specialized region of the nucleus called the nucleolus. So this region is a specialized region of the nucleus, so it's, number one, inside the nucleus, but this is where ribosomal RNA is made, and certainly peptidyl transferase is made here. So here's the key that I want you to understand, okay? Basically, whatever is on the tRNA and the P site, peptidyl transferase catalyzes the transfer of all of that onto the amino acid in the A site, okay? That's a key thing to understand. Now, and actually, let's do that again. What color did I use? So this one was blue. This one was blue. This one was green. For example, let's add the black one here. Let's add the red one. Let's add another blue one. Let's do the same thing over here. So this one was blue. This one was green. Then I have a black one. Then I have a red one. And then I have another blue one. Okay. So, now we have in this picture right here, okay, we have this growing polypeptide chain basically in the A site. Something very important is going to happen here, okay? There's a wedge, okay? Well, what does a wedge do? Let's say I have an object right here. So here's an object kind of sitting on the ground. And let's say I use a wedge like this. So I put a wedge right here, put a wedge right there, okay? What's going to happen? What's going to happen when I put a force on it like that? Let's say I strike that with a hammer. So let's say I strike with a hammer. Force due to a hammer. What's going to happen? Well, if I strike that wedge like that, whatever is this box right here is essentially going to move to the left, right? Hopefully that makes sense to you. So kind of the same thing's going to happen here. I have a protein that's referred to as EFG-GTP. This essentially is the wedge. This is the wedge, okay? You would also have this in eukaryotic cells. So what's essentially going to happen is this wedge is sort of going to come right here. Kind of shade that in. This is the EFG GTP. You need to be familiar with this protein because the same thing's going to apply in eukaryotes. So this is what's essentially going to happen. EFG GTP, while in the GTP bound state, is going to essentially apply a force. It's going to release some energy. So the force, you could effectively think of as being like a hammer hitting on there. And so basically it's going to cause three things to migrate. Okay? So number one, the mRNA is going to migrate this direction. Okay? Whatever's in the P site migrates this direction. Whatever's in the A site migrates this direction. And it turns out that each of these things, the length of migration is one codon, which happens to be 3 nucleotides, right? So if you think about this, okay, if everything migrates over one codon, okay, what happens? Well, whatever is in this A site, right, this growing polypeptide chain that's attached to the tRNA in the A site, notice that it ends up in the P site. Okay? This whole business, this growing peptide chain attached to the tRNA is now in the P site, right? And notice at the end of the last reaction, I of course had this I had this free tRNA. Well, if everything migrates over one codon, then that's going to end up in the E site, right? This is the E site. And when something ends up in the E site, we just say it dissociates. It dissociates, right? And also the mRNA moves over one codon. So this AUG that's right here, let me do this in blue. This AUG that's right here ends up below the E site, okay? Likewise, this sequence of nucleotides, shading these in green, that sequence of nucleotides was basically on the A site. Now it's on the P site, right? And then there would have been, I'll do this in black, there would have been a codon right here, right? Now that codon is in the A site. So hopefully you can see that when that EFG GTP comes in and acts as a wedge, the mRNA, whatever's in the A site and whatever's in the P site, essentially migrate over one codon, in this case to the left. Now, essentially what that does is, from the ribosome's perspective, okay, notice the ribosome essentially moves over one codon in this direction relative to the mRNA. Okay, so essentially what the EFG GTP does is it forces the mRNA over and all the tRNAs, right? But from the ribosome's perspective, essentially the ribosome effectively moved over to the right, even though technically it's everything else that's moving over to the left one codon. But hopefully you see that that growing polypeptide chain that was attached to the tRNA in the A site is now in the P site. Whatever was in the P site, that free tRNA, is now in the E site. It exits and dissociates. And this structure right here, this structure, let me highlight it in light blue. All this business right here, right? All this business minus this incoming amino acyl tRNA, this is how we said we start the cycle, right? Remember, we start the cycle with the growing polypeptide chain attached to the tRNA in the P site. That's essentially how we start the cycle. Hopefully that makes sense to you. So essentially what's going to happen once again is, we have this 2 protein. 2 is going to facilitate the binding of the new incoming amino acyl tRNA into the A site. So the key is that in order to redo a cycle of the ribosome, we have to have the growing polypeptide chain attached to a tRNA in the P site. Okay? And what we're essentially going to do now is look at an analogy that will hopefully make a little bit of sense. Okay? So here's the idea. Okay? I've represented essentially the amino acids as people, okay, and I put the tRNAs as little black circles, or I should say ellipses, with the word tRNA in it, okay? So here's the idea. Let's say that I have, in the P site, we start a cycle, and I essentially have 4 amino acids where this black girl, right, is attached to the tRNA, and on her other side, she's attached to the other people, or the other amino acids. And let's say I have another incoming amino acyl tRNA, right, and this girl's the red girl, and on one side, her hand's holding onto the tRNA, but let's say that she really wants to hold hands with this girl over here. She really wants to hold hands with this girl right here. So what she's going to do with in this case, it's her left hand, she's going to really tightly grab this black girl's right hand. And when she grabs that hand, it effectively causes this tRNA to dissociate, right? I hope that makes sense. So you go from this situation, and if this red girl grabs the black girl's hand, then you end up with this situation, right? Essentially, the red girl is grabbing this whole chain of people right here. This whole chain of people through the black girl are getting translocated onto this red girl. Okay? And so that leaves you a free tRNA in the P site, and then everybody else is attached to the red girl in the A site. So I hope you see how that works. And again, the nice thing is you can rewind it. So we go from this to this. So now the whole polypeptide chain is on the tRNA in the A site, and now we have this free tRNA in the peptidyl site, or the P site. Now, a Tyrannosaurus rex, EFGTP, or EFG-GTP has appeared. So this is EFG-GTP. That's the Tyrannosaurus rex. And everyone will now move over one spot in the ribosome. including the mRNA, right? So remember, the mRNA is below this, right? So the mRNA is also going to move over this direction. And you can imagine, oh no, there's a T-Rex, we'd like to get away from that, right? So everything in the P site is going to move over. He wants to get away from the T-Rex. All this business, all these people with this tRNA, they want to move, so they'll move over one codon into the P site, and so will the mRNA. So all these translocations are essentially going to be one codon or 3 nucleotides, okay? So you see these exclamation marks, I'd like to get over, right? So everybody wants to move over one codon. So everything in the A site moves over to the P site, right? Everything in the P site moves over to the E site. So move over or I'll eat you, right? So everybody wants to get away from the T-Rex, including the mRNA. So the mRNA moves over one codon. So everybody moves over, and so whatever's in the A site ends up in the P site. Whatever's in the P site ends up in the E site. This is the fundamental mechanism of the ribosome right there, and it's catalyzed by EFG-GTP. Okay? Thank God the Marines are here to stop the T-Rex. Okay? So you could sort of think of this is the hydrolysis of GTP. So essentially the hydrolysis of GTP is essentially what causes the T-Rex or the EFG-GTP to leave. Okay? And the net result of that is that you have this group of people, right? This group of people in the P site attached to the tRNA in the P site, and then basically whatever's in the E site, the exit site, they leave, right? That tRNA leaves because it's the exit site. It dissociates, okay? And so you end up with a situation like this. So what I want you to notice is the following. Let me actually go and get this slide right here. I'll copy, copy, move down, and I'll paste right here. Paste, there we go. So I want you to look at the difference between these two. What's the difference between these? Well, here in the P site, what happened? Well, I have 4 amino acids represented by those 4 people, right? Also, what's important to realize is that's 3 peptide bonds because it's 4 amino acids, right? And what did I do? I lengthened it or elongated it by 1 amino acid to where now, oops, let me erase that. Now I have 5 amino acids and 4 peptide bonds, right? And this is a really important point. It's important to understand. The process by which you add amino acids to the growing polypeptide chain from the ribosome's perspective, that process is called elongation. Elongation. And the reason it's called elongation is we're lengthening the length of the polypeptide chain by 1 amino acid per cycle of the ribosome. So hopefully that makes a little bit of sense. Okay? And you'll keep doing this cycle of lengthening the protein by adding amino acids. Okay? You'll keep doing this. And eventually you'll get to something that's really, really long, lots of amino acids, and of course, at the end of the cycle, notice it'll be attached to a tRNA in the P site. Remember, that's how we start every cycle of the ribosome. But this time, something different is going to happen. Let's say that this amino acid, I'll highlight it in black, let's say that amino acid right there, we want to be the terminal amino acid. So what will happen from transcription is we'll get an mRNA with this codon. Okay, this codon right here is UAG. Okay, what I'll do is I'll show you this. So there's UAG right here. Let me do this and I'll highlight it. So these right here, UAG, UAA, and UGA. These are what we call stop codons. And essentially what a stop codon does is it codes for something called a release factor. This right here is the release factor. Okay? So whenever the ribosome encounters a UAG, instead of a tRNA or an aminoacyl tRNA coming into the A site, instead something called a release factor comes in. Okay? And what the release factor essentially does is it catalyzes the hydrolysis off of the protein. So this right here, this bond between the tRNA and the protein essentially is broken. So this is, again, this is the black amino acid that I highlighted there. So essentially that whole polypeptide chain gets hydrolyzed off. And so what essentially happens is now you have a free tRNA, right? This is your free tRNA in the P site. You, of course, have a release factor in the A site. And as soon as that happens, okay, everything dissociates. Okay, so notice, these are not the subunits you'd see in eukaryotes, but the same thing's going to happen. The large subunit dissociates. Small subunit dissociates. mRNA dissociates, and all this stuff, the tRNA, you'll have, of course, ribosomal RNA, release factor, all that stuff dissociates. And then, of course, they will essentially be recycled to do more translation of more mRNA, okay? In which case you'll have an initiation complex that assembles all that back together. But we're not going to talk about that here, okay? And notice the process of elongation was catalyzed by the ribosomal RNA component peptidyl transferase. So hopefully that makes a little bit of sense. In the next video, something that's not required for A&P, but required for biochemistry, we'll look at the mechanism of peptidyl transferase, and we'll find it's actually not that difficult. This process, by the way, where a release factor comes in because of a stop codon and everything dissociates, this process is called termination. So termination is the process by which a release factor catalyzes the hydrolysis off of this entire protein chain right here. And you basically end up generating a carboxyl group at the end, and this is called the C-terminus of the protein. That's not really important, but from a biochemical perspective it is. Okay, so the entire ribosome dissociates, and only once the ribosome reassembles can translation occur again. So hopefully that makes a little bit of sense. Here's the genetic code table. Let's practice figuring out what each codon codes for. So here's UCA. What does UCA code for? Well, here's U, here's C, and then we go into this box right here. We find that UCA codes for SER, which is the 3-letter abbreviation for serine. Okay? What about UGA? Well, here's U, here's G, and then UGA, notice, is a stop codon. UGA is a stop codon, and so it codes for, if we go back to this slide, it codes for a release factor. So this codes for a release factor, okay? And not only would UGA do that, but also UAA and UAG, okay? What about UGG? Let's do that in blue. So here's U, here's G. UGG is tryptophan, okay? So this is TRP, but that's the abbreviation for tryptophan, okay? So hopefully that makes a little bit of sense. So on the test, what I'll do, probably in a multiple choice question, is I'll give you this genetic code table, and I'll give you a codon and say what amino acid does it code for. If it was a stop codon, you would essentially say it codes for a release factor. Okay. The elongation of the polypeptide chain, i.e. transfer of the growing peptide chain from the P site to the A site, in the mechanism of the ribosome is carried out by what enzyme? Okay. Well, there's only one enzyme here we've talked about thus far, which kind of makes it obvious that the answer is peptidyl transferase, and that's the answer. So peptidyl transferase comes from the fact that it's transferring the peptide chain from the tRNA in the P site to the tRNA in the A site, and that's catalyzed by peptidyl transferase. In prokaryotes, the enzyme that acts as a wedge and forces the ribosome to translocate along the mRNA is what? Well, it's EFG-GTP. Okay, so this is a G protein that catalyzes everything to basically move over one codon. So this catalyzed the translocation of one codon. So hopefully that makes a little bit of sense. And from the ribosome's perspective, if you go back to these slides up here, let's find the slide. So if you look at this with respect to the ribosome, the mRNA, things in the A site and things in the P site are essentially moving over to the left in this picture. But from the ribosome's perspective, everything's actually moving. The ribosome's actually moving to the right. Okay, but it's sort of like the ribosome is indirectly moving, but everything inside the ribosome is moving to the left. So indirectly, the ribosome is moving to the right. So hopefully that makes a little bit of sense. Okay. Ribosomal RNA, the catalytic part of the ribosome, if you wanted to be really specific, this catalytic part, the name of this enzyme, like we mentioned in the previous question, is peptidyl transferase. And we mentioned that peptidyl transferase is synthesized in a special part of the nucleus, which is called what? Okay. Number one, it is synthesized by an RNA polymerase. It's still synthesized by an RNA polymerase, but in a special part of the nucleus which is the nucleolus. Okay, that's a very dense part of the nucleus where ribosomal RNA is synthesized. mRNA and transfer RNA are not synthesized there. They're synthesized in the general nucleoplasm of the nucleus. Okay, so hopefully that makes a little bit of sense. In the next video, what we're going to do is we're going to look at the function of the rough ER and the Golgi apparatus. We're going to kind of condense those into one video because their functions are really complementary to one another. We have to put them all in one video to make it all make sense. So hopefully that video helped you understand the ribosome. See you in the next video.